we get experimental science. We're curious, non-judgmental.
It's a birthday microscope stream, Kai, and Baby Alona and I are hanging out together. She is my lovely co-host for part of this evening, right? You're my co-host? Yeah, you're my co-host. But yeah, we're looking under the microscope at 30 million year old amber. Good to go! We're good to go! Alrighty, y'all. The swirls of this make it very pretty, but the swirls also are unfortunate for us to be able to grant. So chat, this animal, this insect is 2 millimeters. 2 millimeters in length. Alrighty, so I'm gonna zoom on out and we're gonna zoom in manually. And Gwen, I think, has already guessed what this might be based on the zoom out. Yeah, it might be something boring. It's okay. You're right. It's Dad's being silly because it's 30 million year old insect. None of it is technically boring. Actually, this is quite a beautiful specimen, Gwen. It looks like there might be a bit of flower in there. It looks like that might be a flower. No, it's a second insect. There's 2 insects actually in here. Right there, those eye cells are really stunning. Let's brighten it up a little bit. We'll take an image like this. She's like, guys, it's beautiful that we can find this. Male, so Tarzan, I don't think it's a sex difference right here. Only because of the structures. I think that thing down there is either a tick or a flea. I think it's a different species. It might be a parasitic mite as well, as Cliff pointed out, given the size. Alright. And we'll color adjust as well and see if it looks any cleaner. Slightly cleaner there as well. So you can actually see, I think, the eye spots quite nicely. And we definitely have a midge in here. The way we can tell that is in turn the abdominal structure and the base of the abdomen. The eyes are also quite stunning. You can see some of the little dots on the eyes. Those are the ocelli, or the eye cells. The antennal structure here, and then this, I think Cliff is right. It may very well be a mite of sorts. Nothing mitey about it. It's probably, given its proximity towards this animal, I don't know if it would have been a parasitic mite. Yeah, this smaller one here, it's a different species, Kanara. This is most likely a mite, and if it's parasitic, that's the only reason why they would be in this close proximity. It was probably latched onto this animal, and it was drinking its blood, or known as the hemolymph in an insect. There'd be no reason why they would be so close in proximity. The other mites that are not parasitic, they're called food mites, and they just feed on whatever food source is readily available. So they'll enter the lab and feed on the fruit fly's food in the same container, and they're very bad to have in the lab. But the likelihood of them being caught in the resin of the amber would be less likely unless it was parasitic. So that is my bet on this one. The small dot. So Kanara, unfortunately, we're at max zoom on the camera. This is what I was looking at, Kanara, is this here. Sometimes we've caught those where there are, in fact, eggs being laid. If it's a last-ditch effort, if they're dying, sometimes one of the things that they do is they just release eggs. As like, I'm gonna die, I need to do something, and this is how I've maybe preserved my genetic line. That's actually how we've caught the fruit flies in amber that have been laying eggs. Those were much easier to tell because the fruit fly egg structure isn't just a nebulous circle, but rather an ovular shape with 2 hairs protruding out of them that are the breathing tubes for those animals and the eggs. That is unfortunately not very visible in these. These mite eggs are just circular. So given the resolution of the camera and what we're looking at, it's very difficult to tell if that's what it is or if it's just schmutz. Does the mite have a requisite number of legs? So, Golganak, it looks like it does have the correct number. And it looks like we can see the mouth parts as well. So the mouth parts being up here and the legs being down here. It does look like it's correct, although I'm seeing some doublets in the image. Like this, if we're zooming in a little better, it looks like this is one leg and not just a doublet occurring, but part of it is just the resolution that we're getting. What do you think, little one? So let's actually flip over the sample and see if we can find any other details. So Golganak, this is a much better angle to see the midge. And actually, I kind of like this image. We'll adjust the image in just a moment, but it looks like, it almost looks like, you know, it's a nice reference for scale and size. You would assume, at least I assumed, midges would be smaller. Oh, come on. Oh, come on, little one. Yeah. Here, you can hang out with Papa a little bit. Let's center this a little bit better. And we'll get a better viewpoint of these two animals. There we go. It's a little bit better centered. So, let me adjust the lighting on this now. We're not seeing as much in terms of detail from this side. You can see that it's actually, the image is a little bit scuffed on the mite, or sorry, the midge, because it's just like stretched here, the image. So, but you can see at least the eye cells. These individual little dots here aren't just pixelations. Those are actually the ommatidia, or the parts of the compound eye. But yeah, this image is unfortunately scuffed. If we zoom out to the top of the amber, do y'all see those scratches? And you see how blurry it is right here. So this is it in focus on the top, and it's just all kinds of scuffed. And so that's distorting the image on the lower focal plane. And then let's go ahead and take an image with it color adjusted. And actually, this one might be a good way to see the eye cells. So there's a pretty good look at the eye cells there. Antenna structure, so that again is our midge, and then our gnat, or not our gnat, the mite below here. Before we wrap up the sample, I want to see if we have any other creatures in here. We've seen these before. These two dots in here. And last time we saw them, we identified them as potential seeds. And so that's my suspicion, is that there's some kind of seeds in here again. There is also an insect here. Part of one. It's much cleaner underneath the scope, but there we go. There's some kind of creature right there. So Risto, it's beautiful, right? This level of all this layering, right? And so the dream would be, Ristarok, that it's some kind of, you know, feather or something like that. But what it actually is, it's discoloration from contamination. So instead of being pure resin, it's instead a mix of different things. In part, it could be a contaminant. In another part, it could be that the temperature and the humidity were different. It's just not solidifying fast enough to be able to be pure in color. And it makes for a beautiful stone, but it makes for a very frustrating image look to it. What we do have here, though, is an insect. It looks like it's split into two, but it very much has a cricket-y body to it. Almost a little bit cockroachy for the lovely Gwendolyn P. But I can tell y'all, there's not enough information here to actually glean what it is. So really, in this sample, we're limited to the mite. Our little mite down here. And our gnat up here. Or our midge, as it were, which is a different subgroup of it. And so that rounds out what we found today. We also took the same image from a different angle to see what else it may look like with the different color contrast. Again, we talked about why the amber looks a particular color, why it has a scuffed variety, and then we talked about the biology behind the insect as well. Really cool insects in the amber as well. I hope you all enjoyed it. Let's